Basics of Medical Biochemistry. First edition, 2022. Chapter 4. Chemistry of Proteins. Part 2 of 2. By Professor Dr. Muhammad Jawad. MBBS, Tau, M.Phil Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. 4.5 Brief detail of some important proteins. 4.5.1 Hemoglobin. HB hemoglobin is a conjugated protein also known as respiratory protein, or metalloprotein. It is composed of globin, a protein part 96%, and heme, iron porphyrin 4%. The concentration of hemoglobin in blood is in adult male equals 14 to 16 g/dl. In adult female equals 12 to 15 g/dl. In infants equals 16 to 18 g/dl. Hemoglobin is a tetramer, each subunit has a heme moiety attached to the polypeptide chain. It is a red-colored protein found inside the RBCs. A single RBC has 29 picograms of hemoglobin. Total amount of hemoglobin in the circulating blood of an adult is about 900 grams. In human beings, following types of hemoglobin are found. IHBA or HbA1, it is the normal hemoglobin in adults, representing 97% of total hemoglobin. It has 2 alpha, and 2 beta chains. 2-HbA2, it is 2.5% in adults. It has 2 alpha, and 2 delta chains. 3-HbF, it is fetal hemoglobin. It has 2 alpha, and 2 gamma chains. The oxygen binding affinity of HbF is higher than that of HbA. For glycated hemoglobin, HbA1c, normally the level of HbA1c is up to 5%, but in uncontrolled diabetes mellitus, HbA1c level may reach up to 12%. In case of hyperglycemia, HbA reacts non-enzymatically with glucose to form glycated hemoglobin or HbA1c. It indicates the presence of hyperglycemia for the last 2 to 3 months. V methemoglobin, hemoglobin M. Normally, blood methemoglobin level is 1 to 2%. In HbM, histidine has been replaced by tyrosine. HbM has Phe3+ rather than Phe2+. Thus, it can neither bind nor transport O2. Normally, the enzyme methemoglobin reductase reduces the Phe3+ of HbM to Phe2+. It is a basic protein, as relatively contains a large proportion of lysine, arginine, and histidine. The hemoglobin of different species is different, only concerning amino acids of globin portion, otherwise, heme portion is same in all types of hemoglobin. The iron content of human hemoglobin is 0.34%. The molecular mass of mammalian hemoglobin is 64.5 kDa. The hemoglobin molecule is composed of 4 globin peptide chains, each combined with a heme group and all held together in a definite arrangement by hydrogen bonds, sulfide linkage, and van der Waals forces of attraction. The hemoglobin molecule undergoes dissociation in solution at pH below 6 and above 9.5. Following 4 kinds of globin peptide chains have been identified. I-alpha globin peptide chain, 2 beta globin peptide chain, 3 gamma globin peptide chain, 4 delta globin peptide chain. Alpha chain contains 141 amino acids while beta, gamma, and delta chain contains 146 amino acids. The amino acid sequence in alpha, beta, and gamma chains is completely known, but in the delta chain, it is partially determined. 4.5.1.1 Structural levels of hemoglobin. 4 levels of structures are observed in the organization of hemoglobin molecule, as follows. I. Primary structure, it is represented by the sequence of amino acids in various chains. 2. Secondary structure. Certain portions of chains are formed into helices and give secondary structure. 3. Tertiary structure. The folding of each chain and relations of various portions of the chains to other portion represents the tertiary structure. 4. Quaternary structure. It is represented by the organization of all 4 chains in the complete hemoglobin molecule. In the beta chain, heme group is found to exist between histidine residues at positions 63 and 92, same in gamma, while in the alpha chain it is in between 87 and 58. Figure 4.13 Structure of hemoglobin molecule, complete molecule, left, iron-containing heme, right. Heme is composed of a protoporphyrin ring and iron, ferroprotoporphyrin. The ferrous iron, Fe2+, of each heme is bound by coordination bonds with nitrogen atoms of the pyrrole ring. Globin is tightly bound to 4 heme groups, prosthetic groups. In the hemoglobin formula, the 4 N atoms of the heme ring are considered to be connected to iron by linkage 1, 2, 3, and 4 in the plane of the paper. Linkage number 5 of iron is attached to the imidazole N of histidine below the plane, while linkage number 6 is above it. In the absence of O2, a water molecule is found at valence number 6 of iron. The oxygenation of hemoglobin involves displacement of this water by O2. H2O.Hb plus O2 HbO2 plus H2O. The overall shape of the hemoglobin molecule is a spheroid having the following dimensions. Length, 64 a. Breadth, 55 a. Height, 50 a. Figure 4.14 Structure of protoporphyrin 9 and heme. The heme groups are in 4 separate pockets on the surface of the molecule. The iron atoms in the neighboring pockets between alpha and beta chains are 25A apart. The iron atoms of alpha chains are 36A and of 2 beta chains are 33.4A apart. In the organization of the hemoglobin molecule, the 4 chains are arranged in the tetrahedral of 2 pairs, alpha-beta, each called a dimer. There is much more area of contact between unlike chains than like chains. That's why dissociation of hemoglobin is in the following way. Hb2 alpha-beta. 4.5.1.2 Binding of oxygen to hemoglobin and myoglobin. Hemoglobin can bind 4 oxygen molecules, 1 at each of its 4 heme groups. There are 2 forms of hemoglobin. I-deoxy or T form, it is also known as a tense form of hemoglobin. It has a low oxygen affinity for hemoglobin. The 2-alpha-beta dimer of deoxyhemoglobin is linked by Pfau ionic and hydrogen bonding. 2-oxy or R form, it is also known as relaxed form. It has O2 bound to heme. The binding of O2 results in the rupture of some of the bonds between its 2 alpha-beta dimers. The oxygen dissociation curve for hemoglobin is sigmoid in shape, while for myoglobin is hyperbolic. The PO2 of 1 mm Hg can achieve half saturation of myoglobin, but for half saturation of hemoglobin, the PO2 needed is 26 mm Hg. The oxygen myoglobin, MbO2, and deoxygenated myoglobin, Mb, exist in simple equilibrium. Mb plus O2 MbO2. This equilibrium is shifted to the right on the addition of O2 and to the left on the removal of O2. This property reflects the O2 storing capacity of myoglobin in muscles at low PO2. The oxygen dissociation curve, sigmoid, indicates the cooperative binding of O2 by its 4 subunits, i.e., the binding of O2 at one heme group increases the O2 affinity for the remaining groups. The O2 and hemoglobin binding is affected by IPO2, through heme-heme interaction. 2 pH of the environment allosteric effectors. 3 PCO2. 4 2,3 bisphosphoglycerate. The oxygen binding with increasing affinity is called heme-heme interaction. It is difficult for the first oxygen to bind with hemoglobin, but subsequent binding is easy and rapid, as shown by the steep upward curve in the region of 20 to 30 mmHg of PO2. The affinity for the last O2 is 300 times more than the first O2 bound. The loading of hemoglobin takes place in the lungs and unloading in the peripheral tissues. 4.5.1.3 Bohr effect. The release of O2 from hemoglobin takes place at low pH while oxygenation of hemoglobin takes place at high pH. These effects shift the oxygen dissociation curve to the right and left respectively, called the Bohr effect. The pH is reduced by an increase in H+ concentration or PCO2. Mechanism of Bohr effect: the O2 is released from the hemoglobin in tissues as PCO2 is high and there is more H+ concentration, but this condition is reversed in lungs where O2 is bound with hemoglobin. Thus, a small change in pH between the lungs and tissues makes hemoglobin a more efficient transporter of O2. The Bohr effect reflects the high affinity of deoxyhemoglobin for proton than oxyhemoglobin. This effect is caused by the ionizable group at the N-terminal alpha amino group and specific histidine side chains. Bohr's effect can be represented by HbO2 plus H plus H.Hb plus O2. Oxyhemoglobin, deoxyhemoglobin. It is clear that an increase in proton, downward arrow pH, shifts the equilibrium to the right, whereas an increase in PO2, downward arrow proton, shifts the equilibrium to the left. Effect of 2,3-bisphosphoglycerate on oxygen dissociation curve for hemoglobin and myoglobin. It is an important regulator of binding of O2 to hemoglobin. It is an intermediate product of glycolysis and is found abundant in RBCs. The binding of 2,3-bisphosphoglycerate reduces the affinity of hemoglobin for O2 and shifts the curve to the right with the release of O2. Factors shifting the oxygen dissociation curve, ODC, to the right. It means that at a given PO2 more oxygen is released from the hemoglobin. It occurs in the following conditions: I upward arrow H+ concentration or low pH. 2 upward arrow CO2 concentration. 3 upward arrow temperature. 4 upward arrow 2,3-bisphosphoglycerate. V upward arrow thyroid hormone. 6 upward arrow metabolic rate, exercise, stress. Factors shifting the oxygen dissociation curve, ODC, to the left. It means oxygenation of hemoglobin at a given PO2. Its causes are I downward arrow H+ concentration or high pH. 2 downward arrow CO2 concentration. 3 downward arrow temperature. 4 downward arrow 2,3-bisphosphoglycerate. V upward arrow carboxyhemoglobin formation. 6 upward arrow methemoglobin and co-poisoning. Figure 4.15 Oxygen dissociation curve, ODC, of hemoglobin. 4.5.1.4 Significance of oxygen dissociation curve, ODC. The sigmoid shape of ODC is of great significance because, by it, the process of oxygenation and deoxygenation is carried out in an optimum way. In the lungs, PO2 is 104 mmHg, which is responsible for full saturation of hemoglobin, about 97%. In the tissues, PO2 is 23 mmHg, which is responsible for the release of O2 from hemoglobin to the tissues. Effect of pH and CO2 on oxygen affinity of hemoglobin. In addition to carrying O2 from lungs to tissues, hemoglobin also carries H+ and CO2, two end products of cellular respiration, from tissues to lungs and kidneys where they are excreted. In the cells of peripheral tissues, organic fuels are oxidized by the mitochondria, using oxygen brought from the lungs by hemoglobin, with the formation of CO2, water and other products. The formation of CO2 causes an increase in H+ concentration, decrease in pH, in the tissues, since the hydration of CO2 yields H+ and HCO3. Carbonic anhydrase. CO2 plus H2OH plus HCO3. The binding of O2 to hemoglobin is influenced by pH and CO2. The binding of H+ and CO2 is inversely related to the binding of O2. At the relatively low pH and high CO2 concentration in tissues, the affinity for O2 is decreased as H2O is bound. Conversely, in the lung capillaries, as CO2 is excreted, blood pH raises, the affinity for O2 increases and protein binds more O2 for transport to tissues. This effect of pH and CO2 on the binding and release of O2 by hemoglobin is called the Bohr effect. Effect of H+ concentration on oxygen affinity of hemoglobin. The oxygen saturation curve of hemoglobin is influenced by the H+ concentration. HbO2 plus H plus H.Hb plus O2. HbO2 is the oxygenated or deprotonated found in peripheral tissues and H.HbO2 is deoxygenated or protonated form found in lungs. When O2 concentration is high as in the lungs, hemoglobin binds O2 and releases proton by favoring a transition to the R state, oxygenated form. When O2 concentration is low as in peripheral tissues, H+ is bound and O2 is released. As the concentration of H+ raises, protonation promotes the release of O2 by favoring a transition to the T state, deoxygenated form. O2 and H+ are not bound at the same sites in hemoglobin. O2 binds to iron atoms of heme, whereas H+ binds to any of several amino acid residues in the protein and can form ionic bonds. These bonds stabilize the deoxy form of hemoglobin, producing a decrease in O2 affinity. 4.5.1.5 Binding of Co2. Most of the Co2 produced in metabolism is hydrated and transported as bicarbonate ions. Co2 plus H2O H2 Co3 H2 Co3 H plus HCO3. Some Co2 is carried as carbamide bound to the unchanged arged amino group of the globin chain, forming carbamoyl hemoglobin, which helps to stabilize the T state because it is charged and forms a hydrogen bond, resulting in decreased affinity for O2. 4.5.1.6 Hemoglobinopathies. It is a group of disorders of hemoglobin caused by the production of structurally abnormal hemoglobin, insufficient synthesis of normal hemoglobin, or rarely both of them. 1. Sickle cell anemia. It is the most common disorder of hemoglobin synthesis, common in the black population, 1:500, in the USA. It is a homozygous recessive disorder. In this type of disorder, the hemoglobin molecule has 2 normal alpha chain and 2 mutant beta chain. In the beta chain, glutamic acid at position No6 is replaced by valine. This altered hemoglobin is known as HBS and the beta chain as beta S. This HBS becomes sickle-like at the places of tissue hypoxia, in minute capillaries. Sickle cells frequently block minor circulation and may rupture and lead to sickle cell anemia. Once the cycle is started, it continues to destroy RBCs, vicious cycle. It is also known as hemoglobin S disease, alpha-2 beta S2. Figure 4.16 normal, left, and sickled RBCs, right. Sickle cell anemia is characterized by lifelong anemia, hemolytic, painful crises, increased susceptibility to infections, poor circulation. In a heterozygote, one gene is normal and the other is the sickle cell gene. A possible selective advantage of the heterozygote is that it is less susceptible to infection by the malarial parasite, Plasmodium falciparum. Affected children are asymptomatic at birth and symptoms appear as the percentage of fetal hemoglobin decreases during the first year of life. Untreated crises increase morbidity and early death. Laboratory findings in sickle cell anemia. Peripheral blood sample revealed severe anemia, 5 to 9 G/DL, which is normocytic and normochromic. Poikilocytosis with sickle cells and target cells. Anisocytosis showed increased red cell distribution with RDW. Nucleated RBCs and polychromasia. Leukocytosis, WBC, 12,000-16,000/MM3, absolute neutrophilia with shift to left. Thrombocytosis is also common and thrombocytopenia occurs during aplastic crises. Electrophoresis on cellulose acetate at a pH of 8.4 shows 85 to 100% HBS and 15% HbF. Other chemistry tests show increased bilirubin, increased lactate dehydrogenase, LDH, and decreased haptoglobin. Diagnostic tests for HBS, sickle cell preparation, sodium metabisulfite is added to the blood. It reduces the oxygen tension and causes sickling of RBCs. It is viewed microscopically. Dithionite tube test. In this test red cells are listed by saponin. Sodium dithionite removes oxygen from the test environment. The HBS polymerizes and forms tactoids, or liquid crystals, and the solution turns turbid. Therapy for sickle cell anemia. Therapy is usually supportive rather than curative. Preventative measures are meant to eliminate conditions that precipitate vaso-occlusion. Lifespan can be significantly increased with early and effective treatment. Studies of natural populations reveal that individuals with sickle cell disease are capable of normal lifespans. Blood transfusion is required during aplastic crises or splenic sequestration. Chronic blood transfusion results in an iron overload of major organs, resulting in increased morbidity. That's why laboratory monitoring is necessary. The use of chelating agents is required to remove excess iron. Hydroxyurea is also used to reduce intracellular sickling. Alternative treatment includes activation of fetal hemoglobin genes and bone marrow transplantation. 2. Hemoglobin C disease. Hemoglobin C is a hemoglobin variant. In this type, lysine is substituted for glutamate at position No.6 of the beta chain. It is a less dangerous disease. Patients with hemoglobin C disease are suffered from chronic hemolytic anemia. These patients are not suffered from infarction crises and need no specific therapy. 3. Hemoglobin SC disease. In this disease, some beta chains are beta S and some are beta C. It is a double heterozygous disease because both beta chains are abnormal. Hemoglobin level is higher in hemoglobin SC disease. Clinical symptoms are similar to sickle cell anemia but milder. Patients generally have painful crises beginning in childhood. Patients remain well and undiagnosed until they are suffered from infarction crises, example after childbirth or surgery. Blood viscosity is higher than in sickle cell anemia, so retinal problems are seen and more severe. Complications are fewer, but splenomegaly is common. 4. Thalassemias. These are a group of hereditary hemolytic diseases. There is a defective synthesis of the alpha or beta globin chain. Thalassemia is caused by a variety of mutations, gene deletion or substitutions. If the alpha chain is affected, excess beta chain is produced. If the beta chain is affected, excess alpha chain is produced. The excess of alpha chains is unstable, they precipitate within the cells and these precipitates bind to the cell membrane, results in membrane damage. Excess beta chains combine to form HBH, 4 beta chains. This type of hemoglobin has high oxygen affinity and thus acts as a poor oxygen transporter. Important clinical findings in thalassemia include anemia, ineffective erythropoiesis, presence of high oxygen affinity hemoglobin, increased extravascular hemolysis, splenomegaly, gallstones, due to increased intravascular and extravascular hemolysis, skeletal abnormalities, pathological fractures and iron toxicity as a consequence of multiple transfusions. Laboratory findings are decreased RBC, hemoglobin, hematocrit microcytic/hypochromic, downward arrow MCV, downward arrow MCH, downward arrow MCHC, increased reticulocyte count, anisocytosis, and poikilocytosis, target cells, nucleated RBCs, increased bilirubin, decreased haptoglobin, increased serum iron and decreased total iron binding capacity, TIBC. I-alpha thalassemia. It is a defect in which the synthesis of the alpha globin chain is decreased or absent. There are 2 alpha genes on each chromosome 16, total of 4 alpha genes in the diploid state. There are several levels of alpha globin chain deficiencies. Mutations can affect one or more of the alpha genes, resulting in 4 levels of severity. If one is defective, the individual is termed a silent carrier because there is no physical manifestation of the disease. If 2 genes are defective, the person has an alpha thalassemia trait and the condition is called alpha thalassemia minor. If 3 genes are defective, the individual has HBH, beta 4, disease or hemolytic anemia. HBH precipitates to form Heinz bodies. If 4 genes are defective, hemoglobin Bart, gamma 4, disease occurs which causes fetal death known as hydrops fetalis. 2. Beta thalassemia. It is a defect in which the synthesis of the beta globin chain is decreased or absent, depending upon the defective genes inhibited. There are only 2 beta globin genes, one on each chromosome 11. The defect is not deletion. Beta plus gene mutation causes a partial block in beta chain synthesis, whereas beta 0 gene mutation results in the complete absence of beta chain production. Over 180 mutations result in partial to complete absence of beta gene expression. There occurs an increase in 2 delta 2, HBA2, and 2 gamma 2, HbF. A beta thalassemia minor, it occurs when the genes inherited are heterozygous, i.e., they inherit only one defective beta-globin chain. Findings are mild to non-existent anemia, microcytosis, hypochromia is not striking, target cells and basophilic stippling. RDW, iron, ferritin, and TIBC are normal. Specific treatment is usually not required. B beta thalassemia major, Cooley anemia, it occurs when the genes inherited are homozygous, i.e., both genes are defective. Symptoms begin to manifest at the age of 6 months. Infants present irritability, pallor, failure to thrive, an enlarged abdomen with brown pigmentation of the skin. Cardiac failure may occur in the first decade of life. Excess alpha chains precipitate that damage red cell membranes and result in intravascular hemolysis. Bone changes, various facial deformities and splenomegaly occur due to extramedullary hematopoiesis. Laboratory findings include hemoglobin as low as 2 to 3 g/dl, markedly microcytic/hypochromic, marked anisocytosis and poikilocytosis. Basophilic stippling and polychromasia. Hemoglobin electrophoresis reveals 90% HbF and increased HbA2. Infants born with beta thalassemia major are severely anemic and require regular transfusions of blood. This treatment can also prove fatal due to iron overload. The definitive treatment available is hematopoietic stem cell transplantation. 4.5.2 Myoglobin, myoglobin, MB, is an iron-containing, globular monomeric protein made up of a single polypeptide chain of 160 amino acid residues. Myoglobin heme prosthetic group center contains an iron ion complexed with a porphyrin called protoporphyrin IX. It is found in the heart and skeletal muscles. It functions as an oxygen storage unit, providing oxygen to the working muscles. Myoglobin stores oxygen in muscles and other cellular tissue, binding one oxygen atom per protein subunit. Myoglobin, in muscle, receives oxygen from the red blood cells and transports it to the mitochondria of muscle cells, where the oxygen is used in cellular respiration to produce energy. Diving mammals such as seals and whales can remain submerged for long periods because they have greater amounts of myoglobin in their muscles than other animals. Clinical aspect: the normal range of serum myoglobin is 0 to 85 nanogram per milliliter, ng/ml. Normal results rule out a heart attack. Abnormal, above 85 ng/ml, results can also be seen in muscular inflammation, myositis. Kidneys are involved in removing the myoglobin from the blood into the urine. When the level of myoglobin is too high, it can damage the kidneys. Myoglobinuria is the presence of myoglobin in the urine. It is usually associated with rhabdomyolysis or muscle destruction. Myoglobin can be considered a cardiac marker. It is highly sensitive as appears in blood earlier than other markers, within 1 to 3 hours. But its diagnostic value in detecting myocardial infarction is limited because myoglobin is also present in skeletal muscle and renal tissues. 4.5.2.1 Functions of hemoglobin and myoglobin. Hemoglobin transports oxygen from the lungs to tissues. Lungs are the loading point of oxygen in hemoglobin because the partial pressure of oxygen here is more, and the unloading point is a tissue because oxygen partial pressure is less here. Hemoglobin is an efficient oxygen carrier because the solubility product of oxygen is quite low in water-based plasma, and hemoglobin increases the solubility many folds. Hemoglobin also transports carbon dioxide back to the lungs and also maintains acid-base balance in the blood, as a buffer. The hemoglobin gives the blood its characteristic red coloration. The hemoglobin acts as a physiologically active catabolite. It plays a crucial role in erythrocyte metabolism. Myoglobin has a strong affinity for binding for oxygen, which makes it able to store it effectively in muscles. Myoglobin helps the body in the starving situation of oxygen, especially in the anaerobic condition. Myoglobin helps in regulating body temperature. Myoglobin can be used as a cardiac marker. 4.5.3 Collagen. Collagen belongs to the family of fibrous proteins. It is quantitatively the most important protein in mammals, making up about 25% of the total protein. It is the major protein comprising the extracellular matrix, ECM. The extracellular matrix is a complex structural entity surrounding and supporting the cells that are found within mammalian tissues. The ECM is composed of structural proteins, collagen, elastin, cell-binding proteins, specialized proteins like laminins and fibronectins, glycosaminoglycans, hyaluronans, chondroitin sulfates, heparan sulfates, and keratin sulfates, and proteoglycans. There are many types of collagens, about 25 distinct types, that are encoded by many different gene types. Types I, II, and III are the most abundant and form fibrils of similar structures. Type 4 collagen is a major component of the basal lamina, basement membrane. 4.5.3.1 Structure of collagen. The structure of type I collagen revealed that it is composed of 3 coiled subunits, 3 polypeptide chains. Each chain consists of about 1,000 amino acids twisted around each other in a characteristic right-handed triple helix. There are 3 amino acids per turn of the helix and every third amino acid is a GLY, glycine. Collagens are also rich in proline and hydroxyproline. Thus, collagen is a polymer of GLYXY, repeats, where X and Y may be proline or hydroxyproline, or any other amino acids. Type I collagen contains approximately 33% glycine and 21% proline and hydroxyproline. As glycine is the smallest amino acid, it can easily fit into the interior axis of the helix, where there is no space for a larger side group than glycine's single hydrogen atom. For the same reason, the rings of the proline and hydroxyproline are kept outward. These 2 amino acids also thermally stabilize the triple helix of collagen. The tropocollagen subunit is a rod about 300 nanometers long and 1.5 nanometers in diameter. There are some covalent cross-linkages within the triple helices, and a variable amount of covalent cross-linking between tropocollagen helices, to form the different types of collagens found in different mature tissues. Collagen fibrils are semicrystalline aggregates of collagen molecules. Collagen fibers are bundles of fibrils. Type I collagen gives bone its tensile strength. Figure 4.17 Structure of collagen. Source: vectorstalk.com/280432245. 4.5.3.2 Synthesis of collagen. Collagens are principally synthesized by fibroblasts, cells found in interstitial connective tissue like chondrocytes and osteoblasts, muscle cells, and epithelial cells. Its synthesis occurs both intracellularly and extracellularly. Most collagens are synthesized similarly, but the given process is typical for type I and can be described in the following stages. 1. Inside the cell. I3 peptide chains are formed, 2-1 and 1-2 chains, in ribosomes along the rough endoplasmic reticulum, rare. These peptide chains, known as pre-procollagen, have registration peptides on each end. A signal peptide is also attached to each one of them. 2 peptide chains are sent into the lumen of the rare. 3 signal peptides are cleaved inside the rare and the chains are now known as procollagen. 4 selected lysine and proline are hydroxylated inside the lumen. This process needs ascorbic acid, vitamin C, as a cofactor. The hydroxylation of lysine and proline is carried out by the enzymes lysyl hydroxylase and prolyl hydroxylase respectively. V-glycosylation of specific hydroxylysine occurs with glucose and galactose. 6,3-pro-collagen assembled to triple helical structure inside the rare. Intrachain and interchain disulfide bonds are formed at the C-terminal propeptide extension. 7. Procollagen is transported to the Golgi apparatus, where it is packaged and secreted by exocytosis. 2. Outside the cell. The N-terminal and C-terminal propeptides are cleaved by procollagen peptidases, post-translation modifications, and tropocollagen is formed. 2. Multiple tropocollagen molecules self-assembled to form collagen fibrils and multiple collagen fibrils form into collagen fibers. Formation of cross-links in collagen. Hydroxyproline residues are involved in hydrogen bond formation that helps to stabilize the triple helix, whereas hydroxylysine residues are the sites for the attachment of disaccharide moieties, galactose or glucose. The side chains of lysine residues may be oxidized to form the aldehyde, i.e., al-lysine by the enzyme lysyl oxidase. These aldehyde residues produce covalent cross-links between collagen molecules. An al-lysine residue on one collagen molecule reacts with the amino group of a lysine residue on another molecule, forming a covalent Schiff base that is converted to more stable covalent cross-links. Aldol condensation also may occur between two al-lysine residues, which forms the structure lysine-or-lysine, lysine-or-lysine. Finally, collagen is attached to cell membranes via several types of protein, including fibronectin and integrin. Figure 4.18 Biosynthesis of collagen. 4.5.3.3 Degradation of collagen. Normal collagens are highly stable molecules, having long half-lives of several months. But connective tissue is in a dynamic state and is constantly being remolded, often in response to growth or damage of the tissue. Collagen degradation in the extracellular matrix is accomplished by a family of collagenases that cleave intact collagen fibers into smaller fragments that can be phagocytosed and further degraded by lysosomal enzymes to their constituent amino acids. 4.5.3.4 Collagen diseases. Following are the important collagen diseases. 1. Ehlers-Danlos syndrome, EDS. It is also known as Cooties hyperelastic, is a group of inherited connective tissue disorders, caused by a defect in the synthesis of collagen. Connective tissue helps support the skin, muscles, ligaments, and organs of the body. Depending on the individual mutation, the severity of the syndrome can vary from mild to life-threatening. Symptoms vary widely based on which type of EDS the patient has. In each case, however, the symptoms are ultimately due to faulty or reduced amounts of type 3 collagen. EDS most typically affects the joints, skin, and blood vessels, with symptoms such as loose, overly flexible joints, smooth or stretchy, easily bruised skin, abnormal wound healing and scar formation, and small, fragile blood vessels. All forms of EDS affect the joints, causing hypermobility or joints that extend beyond the normal range of motion. As a result of their hypermobility, individuals with EDS are more susceptible to injuries such as dislocations, subluxations, sprains, strains, and sometimes fractures. Because it is often undiagnosed, some instances of EDS have been mischaracterized as child abuse. There is no known cure. Treatment is supportive. Figure 4.19 Individual with Ehlers-Danlos syndrome displaying hypermobile joints. Source: practicalpainmanagement.com. 2. Osteogenesis imperfecta. It is also known as brittle bone disease, or Lobstein syndrome, is a genetic bone disorder. People with osteogenesis imperfecta are born without the proper protein, collagen, or the ability to make it, usually because of a deficiency of type I collagen. This deficiency arises from an amino acid substitution of glycine by cysteine in the collagen triple helix structure. The larger cysteine molecule creates sterically interruption that creates a bulge in the collagen complex. Glycosylation of the cysteine molecule promotes further interference within the structure. As a result, the body responds by hydrolysing the improper collagen structure. As collagen is an important protein in bone structure, this impairment causes those with the condition to have weak or fragile bones. As a genetic disorder, osteogenesis imperfecta is an autosomal dominant defect. Most people with osteogenesis imperfecta receive it from a parent, but it can also be an individual, de novo or sporadic, mutation. A disorder of tooth development as dentinogenesis imperfecta may also be seen in osteogenesis imperfecta. 3. Alport syndrome. It is a genetic disorder characterized by glomerulonephritis, end-stage renal disease and hearing loss. It can also affect the eyes. Hematuria is almost always found in this condition. Alport syndrome is caused by mutations in collagen synthesizing genes. Mutations in any of these genes prevent the proper production or assembly of the type 4 collagen network, which is an important structural component of basement membranes in the kidney, inner ear, and eye. The affected basement membranes of the kidneys are not able to filter waste products from the blood and allow blood and protein into the urine. There is gradual scarring of the kidneys, eventually leading to renal failure. Gene therapy is a possible treatment option. 4.5.4 Elastin. Elastin is a connective tissue protein that is responsible for properties of extensibility and elastic recoil in tissues. It is also known as elasticin. It is a protein that coils and recoils like a spring within the elastic fibers of connective tissue and accounts for the elasticity of structures such as the skin, blood vessels, heart, lungs, intestines, tendons, and ligaments. Elastin functions in connective tissue together with collagen. Whereas elastin provides elasticity, collagen provides rigidity to connective tissue. Elastin is normally no longer made after puberty and aging begin. It is synthesized as a monomer, one polypeptide chain, 70 keta, called tropoelastin. Some of the prolines are hydroxylated by prolyl hydroxylase. It is not synthesized in a preform with extension peptides. There is the absence of repeat GLYXY sequences, triple helical structure and carbohydrate moieties. After secretion from the cell, certain LYSYL residues of tropoelastin are oxidatively deaminate to aldehydes, allycine, by LYSYL oxidase, the same enzyme as found in collagen. The major cross-links formed in elastin are the desmosine. Once cross-linked in its mature, extracellular form, elastin is highly insoluble and extremely stable and has a very low turnover rate. Figure 4.20 Structure of elastin. Table 4.2 Major differences between collagen and elastin. S.No. Collagen. Elastin. 1. Different genetic types. Only one genetic type. 2. The main organization is a triple helix. No triple helix, random coil. Organizations permitting stretching and elasticity. 3. GLYXY, and repeating unit. No, GLYXY, and repeating unit. 4. Hydroxylysine is present. Hydroxylysine is absent. 5. Carbohydrate is present. Carbohydrate is absent. 6. Intramolecular aldol. Cross-links are found. Intramolecular desmosine. Cross-links are found. 7. Extension peptides are seen. During biosynthesis. Extension peptides are not found. During biosynthesis. 8. Rigid in nature. Elastic in nature. Role of 1-antitrypsin in elastin degradation. Blood and other body fluids contain a protein called 1-antitrypsin, which inhibits several proteolytic enzymes, proteases or proteinases, that hydrolyze and destroy the protein. Most of the 1-antitrypsin of the plasma are synthesized and secreted by the liver. The remainder is synthesized by several other tissues. Role of 1-antitrypsin in lungs. In the normal lung, the alveoli are chronically exposed to low levels of neutrophil elastis released from activated and degenerating neutrophils. The presence of 1-antitrypsin makes it possible to protect elastin of alveolar walls from the degrading action of elastis. Emphysema due to 1-antitrypsin deficiency. In the United States, approximately 2 to 5% of the patients with emphysema are predisposed to disease by inherited defects in 1-antitrypsin. This disorder affects about 1 in 1,500 to 3,500 individuals with European ancestry. It is uncommon in people of Asian descent. The deficiency of 1-antitrypsin is caused by mutations in the SERPINA1 gene. This gene is responsible for the production of 1-antitrypsin. The 1 carrier is a person who has one normal 1 gene, M, and one defective 1 gene, usually Z or S. It is believed that over 19 million people in the United States are carriers. Most 1 carriers are MZ or MS. Smokers are vulnerable to getting emphysema because cigarette smoking oxidizes the 358th methionine residue of 1-antitrypsin. The 1-antitrypsin with oxidized methionine is powerless to exhibit its inhibitory activity against elastis. The deficiency of elastis inhibitor can be reversed by weekly intravenous administration of 1-antitrypsin. 4.5.5 Fibronectin. It is a major glycoprotein of the ECM. It is a type of cell adhesion protein that mediates the interaction between cells and the ECM. It consists of two identical subunits, two polypeptide chains, each of about 230 kDa, joined by two disulfide bridges near their carboxyl terminals. Each subunit contains a series of globular domains with binding sites specific for particular extracellular components like collagen, fibrin, heparin, heparan sulfate, hyaluronic acid, etc. Fibronectin contains an ARG-GLYASP sequence that binds to the receptor in cell surfaces. Functions of fibronectin are: I. It functions to connect cells together and to ECM via integrin receptors, transmembrane proteins connecting ECM to cellular actin microfilaments. 2. Cellular migration during development, wound healing, regulation, differentiation and growth. 3. Promotes cell adhesion, has a role in thrombosis and blood clotting. 4. Enhances phagocytosis. The interaction of fibronectin with its receptor provides one route whereby the exterior of the cell can communicate with the interior and thus affect cell behavior, and this plays an important role in the adhesion of cells to the ECM. Loss of adhesion protein capability can lead to either physiologic or abnormal cell movement. 4.5.6 Plasma proteins. Human plasma proteins are a mixture of simple proteins such as albumin and conjugated proteins like glycoproteins, lipoproteins, etc. Plasma proteins are regarded as the chief solids of plasma, comprising about 7.0 to 9.0 g/dl. Table 4.3 Normal values of plasma proteins. Type: by precipitation. G/dl. By paper electrophoresis. Percent of total proteins. Albumin 3.6-5.2. 50-70. Globulins 1.8-3.6. 29.5-54. Alpha-1 globulin 0.1-0.4. 2.0-6.0. Alpha-2 globulin 0.4-0.8. 5.0-11.0. Globulin 0.5-1.2. 7.0-16.0. Globulin 0.7-1.5. 11.0-22.0. Fibrinogen 0.2-0.4. 4.5.6.1 Albumin. It is a simple protein with a single polypeptide chain, the most abundant and homogeneous protein. The molecular weight is 69,000. It is composed of 585 amino acids, with 17 interchain disulfide links. It is half of the total plasma protein concentration. It has low isoelectric pH, pi 4.7. Its rate of synthesis is 14 grams per day. Functions of albumin are: contributes 70 to 80% of osmotic pressure, exerts low viscosity, plays an important role in the exchange of water, helps in the transport of several substances like free fatty acids, FFA, Ca2+, bilirubin and steroid hormones. Undergoes constant exchange with the albumin present in extracellular spaces of muscles, skin, and intestine. Clinical aspect: albumin is decreased in severe protein-calorie malnutrition, liver diseases like cirrhosis of the liver and nephrotic syndrome. Hypoalbuminemia leads to edema formation. Edema occurs when total proteins fall below 5.0 g% or when the albumin level falls below 2.5 g%. 4.5.6.2 Globulins. Molecular weight of globulins ranges from 90,000-13,000,000. They can be separated into different fractions as alpha-1 globulins, alpha-2 globulins, globulins, and gamma globulins. The alpha and globulins are synthesized in the liver, whereas gamma globulins are synthesized by plasma cells and B cells of lymphoid tissues. I-alpha globulins, these are glycoproteins, further divided into alpha-1 and alpha-2 globulins. Alpha-1 globulins of clinical importance are alpha-1 acid glycoprotein, alpha-1 beta-globulin, alpha-1 globulins inhibitors, alpha-1 antitrypsin, etc. These are consistently rising in acute and chronic inflammatory conditions, cirrhosis of the liver and malignant conditions. These are also used as tumor markers. Alpha-1 acid glycoprotein is also known as orozomucoid protein. It contains 41% carbohydrates. It is involved in the transport of progesterone and acts as a carbohydrate supplier to the site of injury. Common examples of alpha-2 globulins are ceruloplasmin, haptoglobin, HP, etc. Although ceruloplasmin is not involved in copper transport, 90% of total body copper is contained in it. Ceruloplasmin is increased in pregnancy, inflammation, malignancies, and women using contraceptive pills. Haptoglobin minimizes the loss of hemoglobin via urine. Combining the power of haptoglobin with hemoglobin varies with different phenotypes. 2 globulins, common examples are low-density lipoproteins, LDL, transferrin, C-reactive protein, CRP, hemopexin, etc. Transferrin is a non-heme iron-containing protein. It is a glycoprotein with a molecular weight of 70,000. Its normal concentration in blood is 1.8 to 2.7 g/L. It has an iron binding capacity of 3.4 mg/L. Normally it is 33% saturated with iron. Its level is increased in iron deficiency anemia. The concentration of CRP equals 1 mg/dl. It precipitates with group C polysaccharide of pneumococcus. It can bind heme and bears some antigenic relations. It can bind to T lymphocyte and activate complement. It is regarded as a sensitive indicator of an early phase of inflammation. 4.5.6.3 Immunoglobulins, IGs. These are a heterogeneous families of serum proteins. These are glycoprotein, composed of 82 to 96% polypeptide and 4 to 18% carbohydrates, molecules produced by plasma cells, also known as antibodies and gamma globulins. They constitute 25 to 30% of total serum proteins and are also present in tissue fluids and mucosal surfaces. All antibodies are immunoglobulins, but all immunoglobulins may not be antibodies. They act as a critical part of the immune response by specifically recognizing and binding to particular antigens, such as bacteria or viruses, and aiding in their destruction. The introduction of antigens in the body triggers the appearance of IGs. Basically, all IGs are composed of 4 polypeptide chains, 2 identical light and 2 identical heavy chains linked by disulfide bonds. Light chains are similar in all immunoglobulins and they occur in 2 varieties kappa and lambda. Light and heavy chains are subdivided into the variable and constant regions. Each heavy and light chain contains an amino terminal in the variable region and a carboxy terminal in the constant region. The Fab portions of the antibody determine its specificity and enable the binding of antigen, while the Fc portions are concerned with the biological activity of the antibody. Figure 4.21 General structure of immunoglobulin. Heavy chains are structurally and antigenically distinct for each class. Each immunoglobulin peptide chain has intrachain disulfide bonds to form loops. Each loop is compactly folded to form a globular structure domain. The light chain contains a single variable domain, VL, and a single constant domain, CL. Both of these domains are composed of 110 amino acids. The heavy chain contains one variable domain, VH, and 3 constant domains, CH1, CH2, CH3. The VH domain is composed of 110 amino acids while the CH domain is composed of 330 to 440 amino acids. The hinge region is the segment in the heavy chain, between CH1 and CH2. The hinge region is the site at which the arms of the antibody molecule form AY. It is called the hinge region because there is some flexibility in the molecule at this point. Carbohydrates are attached to the CH2 domain in most immunoglobulins. However, in some cases, carbohydrates may also be attached at other locations. There are 5 immunoglobulin classes, isotypes, of antibody molecules found in serum: IgG, IgM, IgA, IgE, and IgD. They are distinguished by the type of heavy chain they contain. IgG molecules possess heavy chains known as gamma chains, IgMs have mu chains, IgAs have alpha chains, IgEs have epsilon chains, IgDs have delta chains. Immunoglobulin G, IgG. It is the most abundant class of immunoglobulins in the serum. It constitutes 80% of total immunoglobulin. IgG is present in blood, plasma, and tissue fluids. It contains fewer carbohydrates than other immunoglobulins. Its half-life is 23 days, the longest of all of the other immunoglobulins. It can cross the placenta and provide natural immunity to fetus and neonates at birth. Macrophages, monocytes, polymorph nuclear neutrophils, PMNs, and some lymphocytes have Fc receptors for the Fc region of IgG. Not all subclasses bind equally well. IgG2 and IgG4 do not bind to Fc receptors. The consequence of binding to the Fc receptors on PMNs, monocytes, and macrophages is that the cell can now internalize the antigen better. The term opsonin is used to describe substances that enhance phagocytosis. IgG is a good opsonin. The binding of IgG to Fc receptors on other types of cells results in the activation of other functions. It acts against bacteria and viruses by opsonization and by neutralization of toxins. It also activates complement by the classical pathway. Subclasses of IgG are IgG1, IgG2, IgG3, and IgG4. The function of subclasses, IgG1, IgG3, and IgG4 cross the placenta and protect the fetus. IgG3 activates complement. IgG1 and IgG3 bind to the Fc receptor on phagocytic cells, monocytes, and macrophages and mediate opsonization. 2. Immunoglobulin A, IgA. It constitutes 10 to 15% of total immunoglobulins. It is found in milk, saliva, tears, and mucus of the respiratory tract, digestive tract and genitourinary tract. In the serum, IgA exists as a monomer. In external secretions, it exists as a dimer called secretory immunoglobulin. It has a J chain and secretory piece. Its half-life is 6 to 8 days. Formation of secretory IgA. Dimeric IgA binds to the receptor on the surface of the epithelial cells, then it is endocytosed and transported across the cell to the luminal surface. After reaching the surface, the poly-Ig receptor is cleaved. The portion of the receptor that remains attached to the IgA dimer is the secretory component. The secretory piece protects IgA from digestive enzymes and denaturation by bacterial proteases. Figure 4.22 Types of immunoglobulins. Source: examrace.com. Functions of IgA. It provides local immunity. Secretory IgA binds to surface antigens of microorganism and prevents its attachment and invasion of the mucosal surfaces of the respiratory and digestive tract. Immune elimination. Secretory IgA provides an important line of defense against Salmonella, Vibrio cholerae, Neisseria gonorrhoeae, influenza virus, and poliovirus. Secretory IgA present in breast milk protects newborns during the first months of life. It also activates the complement system by the alternative pathway and promotes phagocytosis and intracellular killing of microorganisms. 3. Immunoglobulin M, IgM. These account for 5 to 10% of total serum proteins. Each IgM is a pentamer, i.e., a polymer of 5 monomeric units. These units are held together by disulfide bonds and the J chain. Its mole weight is 900,000, highest of all immunoglobulins, and half-life is 5 days. Most IgM, 80%, are intravascular and also present in low concentrations in intercellular tissue fluids. They cannot cross the placenta, thus the presence of IgM antibody in the serum of newborns indicates congenital infection. It is the earliest immunoglobulin to be synthesized by the fetus, at the age of 20 weeks, and the first immunoglobulin to be produced in the primary response to antigen. It is the antibody that developed against IgG in rheumatoid arthritis as a rheumatoid factor. Monomeric IgM appears on the surface of unstimulated B lymphocytes and acts as receptors for antigens. Functions of IgM: it agglutinates bacteria, activates complement by classical pathway, and causes opsonization and immune hemolysis. It is believed to be responsible for protection against blood invasion by microorganisms. IgM antibodies are very efficient in leading to the lysis of microorganisms. For immunoglobulin E, IgE, its structure is similar to IgG, also known as a reagenic antibody. It has 4 constant region domains. Its mole weight is 190,000 and half-life is 2 days. It is heat labile, being inactivated at 56 degrees Celsius in 1 hour and found mostly in the extracellular region. A normal serum concentration is 0.3 g/ml and it does not cross the placenta. It is produced in the lining of the respiratory and the intestinal tract. It neither activates complement nor agglutinate antigens. It binds to the Fc receptors on the membranes of blood basophils and tissue mast cells. Degranulation of cells occurs when 2 IgE molecules on the surface of these cells are cross-linked by binding of the same antigen. It can mediate immediate hypersensitivity reaction and causes the release of histamine and pharmacological mediators of anaphylaxis from the cell. It is responsible for symptoms of anaphylactic shock, hay fever, and asthma. It plays a role in immunity against helminthic parasites. Since serum IgE levels rise in parasitic diseases, so the measurement of IgE levels is helpful in the diagnosis of parasitic infections. Eosinophils have Fc receptors for IgE, and the binding of eosinophils to IgE-coated helminths results in the killing of the parasite. The physiological role of IgE appears to be protection against pathogens by mast cell degranulation and release of inflammatory mediators. V. Immunoglobulin D, IgD. Its structure is also similar to IgG. Its molecular weight is 175,000 and its serum concentration is 30 g/ml. It constitutes 0.2% of total immunoglobulins, having a half-life of 3 days. IgD along with IgM is the major membrane-bound immunoglobulin on unstimulated B lymphocytes and acts as recognition receptors for antigens. Polyclonal antibodies, or antisera antibodies that are derived from different B cell lines. They are a mixture of heterogeneous immunoglobulins secreted against a specific antigen, each recognizing a different epitope of a single antigen. These antibodies are typically produced by immunization of a suitable mammal, such as a mouse, rabbit, or goat. Larger mammals are often preferred as the amount of serum that can be collected is greater. An antigen is injected into the mammal. This induces the B lymphocytes to produce IgG immunoglobulins specific for the antigen. This polyclonal IgG is purified from the mammal's serum. Monoclonal antibodies, these are derived from a single cell line. These are monospecific antibodies that are identical because they are produced by one type of immune cell that are all clones of a single parent cell. They are designed to bind to antigens that are generally more numerous on the surface of cancer cells than healthy cells. A monoclonal antibody is made so that it binds to only one substance. Monoclonal antibodies are being used to treat some types of cancer. They can be used alone or to carry drugs, toxins, or radioactive substances directly to cancer cells. This has become an important tool in biochemistry, molecular biology, and medicine. Table 4.4 Important features of immunoglobulins. Feature: IgG, IgA, IgM, IgE, IgD. Structure: monomer, monomer, or dimer, monomer, or dimer, monomer, monomer. Type of H chain: gamma, E, delta. Molecular weight: times 1000, 150, 170 or 4002, 900, 190, 180. Serum concentration: mg/dl 1000, 200, 120, 0.05. 3. Fraction of total immunoglobulin in serum, percent: 75, 15, 9, 0.004, 0.2. Found in secretions. Plus ability to cross placenta, plus or 4.5.6.4 Functions of plasma proteins. 1. Transport. Albumin is actively involved in the transport of bilirubin, free fatty acids, Ca2+, Cu2+, Zn2+, steroids, other hormones and a large number of drugs. Lipoproteins transport lipids, hemopexin transport heme, ceruloplasmin transport Cu2+, corticosteroid binding globulin, transcordon, transports cortisol, sex hormone binding globulin transports testosterone and estradiol. Similarly, thyroid binding globulin transport T4 and T3. Transferrin transports iron and haptoglobin binds and transport extracorpuscular hemoglobin. 2. Protease inhibitors, antiproteases like alpha-1 antitrypsin, antichymotrypsin, alpha-2 macroglobulins, antithrombin, etc. 3. Blood coagulation. Many factors are involved in the clotting mechanism and prevent the loss of the excessive amount of blood. Example clotting factors 9, 8, fibrinogen, etc. 4. Anticoagulant activity. Thrombolysis. Plasmin breaks down thrombin and dissolves the clot. 5. Enzymes, pepsin, lipase, cholinesterase, aminotransferases as well as coagulation factors. 6. Hormones, erythropoietin released from kidneys. Although many protein hormones like insulin and glucagon are circulating in the blood but, they are not considered true plasma proteins. 7. Role in inflammatory reactions, C-reactive protein, acute phase response proteins, and orozomucoid protein, alpha-1 acid glycoprotein. A high level of CRP in the blood is a marker of inflammation. It can be caused by a wide variety of conditions, from infection to cancer. High CRP levels can also indicate that there's inflammation in the arteries of the heart, which can mean a higher risk of a heart attack. The orozomucoid is used in the diagnosis and monitoring of inflammatory bowel disease. 8. Role of alpha-1 fetaprotein, AFP. It is a protein made in the liver of a developing baby. AFP levels are usually high when a baby is born, but fall to very low levels. High levels of AFP in adults who aren't pregnant usually indicate certain types of liver disease, cirrhosis or chronic acute hepatitis. An extremely high level of AFP in the blood, greater than 500 ng/ml, could be a sign of liver tumors. High levels of AFP may mean other cancers, including Hodgkin disease, lymphoma, and renal cell carcinoma, kidney cancer. 4.5.6.5 Bence-Jones protein. It is a monoclonal globulin found in the urine, with a molecular weight of 22 to 24 kDa. When the urine is heated, the Bence-Jones protein precipitates between 40C60C and redissolves on further heating to 100C. The precipitate appears again between 40C60C on cooling. It is commonly seen in multiple myeloma and sometimes in leukemia. Summary. The term protein is derived from the Greek word, proteios, meaning in the lead or standing in front or primary or holding first place. Proteins are nitrogenous colloidal substances made up of amino acid residues joined together by a peptide linkage. 1 gram of protein per kg per day is sufficient. Each gram of protein furnishes 4.1 kilocalories of energy. All enzymes are protein in nature, without them life is impossible. All amino acids possess common structural features, including an alpha carbon to which an amino group, a carboxyl group, and a variable side chain are bonded. The form of amino acid having no net charge is known as zwitterion. Essential amino acids cannot be formed in the body. Examples are lysine, leucine, isoleucine, methionine, phenylalanine, tryptophan, threonine and valine. The peptide bond is formed when the alpha-carboxyl group of one amino acid forms a covalent bond with an alpha amino group of another amino acid by the removal of a molecule of water. Simple proteins on hydrolysis give only amino acids. Examples are albumin, globulins, protamines, hist-1s, etc. Nucleoproteins are composed of simple basic proteins, protamines and hist-1s, with nucleic acid, DNA and RNA. Fibrous proteins have an axial ratio of more than 10. Examples are keratin, elastin, myosin and collagen. Globular proteins have an axial ratio of less than 10, they are less stable than fibrous proteins. Examples are albumin, globulin, and ribonuclease. Collagen is composed of 3 coiled subunits, 3 polypeptide chains, as a triple helix. Each chain consists of about 1,000 amino acids. There are 3 amino acids per turn of the helix and every third amino acid is glycine. Elastin is a connective tissue protein that is responsible for properties of extensibility and elastic recoil in tissues. Fibronectin is a major glycoprotein of the ECM. It is a type of cell adhesion protein that mediates the interaction between cells and the ECM. First-class proteins are also known as a complete protein. These are mainly obtained from animal sources like meat, fish, dairy, and poultry products. They are full of the essential amino acids. Primary structure of protein describes the unique sequence of amino acids regarding their types, total number, and arrangement in a polypeptide chain. Secondary structure of protein describes the folding and coiling of peptide chain giving a three-dimensional structure. Hemoglobin is a conjugated protein. It is composed of globin, a protein part 96%, and heme, iron porphyrin 4%. Bohr effect: the release of O2 from hemoglobin takes place at low pH while oxygenation of hemoglobin takes place at high pH. The sigmoid shape of ODC is of great significance because, by it, the process of oxygenation and deoxygenation is carried out in an optimum way. Sickle cell anemia is one of the types of hemoglobinopathies with mutant beta chain. In the beta chain, glutamic acid at position No6 is replaced by valine. Thalassemias are a group of hereditary hemolytic diseases. There is a defective synthesis of alpha or beta globin chain. Immunoglobulins are a heterogeneous family of serum proteins. These are also known as antibodies and gamma globulins. Serum IgE levels rise in parasitic diseases, so the measurement of IgE levels is helpful in the diagnosis of parasitic infections. References. Exercise questions. For MCQ 1-5, see Part 1 of 2 of this chapter. MCQ 6. A 9-year-old boy is presented with hypermobile, loose and unstable joints that dislocate easily. Physical examination suspect that the patient is suffering from Ehlers-Danlos syndrome. Which of the following types of collagens is deficient in this patient? Type I, Type 2, Type 3, Type 4, Type V. MCQ 7. In osteogenesis imperfecta, brittle bone disease, the defect in collagen is due to the substitution of glycine by which of the following amino acids? Alanine, arginine, cysteine, leucine, lysine. MCQ 8. Which of the following is a common feature of collagen, elastin, and fibronectin? All are catalytic protein. All are fibrous protein. All are globular protein. All are metalloprotein. All are plasma protein. MCQ 9. A 10-year-old girl suffering from the brittle bone disease is presented with symptoms of discoloration of the eye sclera, barrel-shaped chest, curved spine, triangle-shaped face, loose joints, and muscle weakness. Which of the following types of collagens is deficient in this patient? Type I, Type 2, Type 3, Type 4, Type V. MCQ 10. A premature baby boy delivered at the gestational age of 30 weeks, died within 24 hours due to acute respiratory distress syndrome. Relevant biochemical investigations and studies on lung tissue revealed that he has deficient synthesis of a protein with abnormality in intramolecular desmosine cross-links. Which of the following proteins is deficient in this patient? Albumin, collagen, elastin, fibronectin, thrombin. Please write answers in the comments. End of Part 2 of 2 of Chapter 4. Thank you.